And yeah, it's a cool day for me to get to talk about phages because our lab actually had our very first phage treatment go all the way from hunting for the phages to prepping them in our lab to getting FDA and IRB approval. And yesterday, a patient at the UC Irvine Medical Center received a phage to treat a MRSA sinusitis infection. And you know, in a way, it's like might have been like 15 years I've thought about doing that for. And it was big thanks to a student I'm going to tell you about. And actually, Daria Van Tine is here in the room. She played a really big role. And yeah, thank you. And Dr. Sage Dunham is a postdoc in our group. He's here too. And anyway, but I'm actually going to start us out. Well, first of all, introduce, you know, where I'm from, which is UC Irvine. We're between Los Angeles and San Diego in Southern California. And we have several microbiome related events going on in the near future. So if you're in the area, please feel free to ask me about it. And before I get too far, I definitely want to acknowledge everybody who contributed to our work and supported our work. And really, I have a poem to share with you, which I have never done before, but my mind was blown earlier this summer when I learned that Amanda Gorman wrote a poem about the microbiome. So Call Us What We Carry is about the microbiome. That's what we carry. And so I owe a big hat tip to Vanessa Hale of Ohio State, who told me this. And so I want to share this really amazing poem, which surprised me so much. So from Call Us What We Carry. At times, over half of our bodies are not our own. Our persons made vessel for non-human cells. And I'm just going to skip to the microbiome part because that's the good part. A human microbiome is all the writhing forms on and inside this body, drafted under our life. We are not me, we are we. Call us what we carry. And I love this concept. It gets to the idea of the holobiont, which refers to not just your own genome, but the exposures you've had to microbes in your life, the microbes that colonize your body, and beyond that, the microbes that you're exposed to in your environment, even if they only transiently have contact with you. And so anyway, I've always loved this concept, and I love how Amanda Gorman described this in her poem, which I had no idea was actually about the microbiome. And so this is a talk about phages. And you know, the main differences between holobionts are actually the viruses. So most of the genetic differences between you and the person sitting next to you are encoded in the microbes on your body. And the phages are like the cutting edge of that. They're the ones that can change the most quickly. They're the ones that can carry new genes into the system very easily. And so they really are a very interesting part of our holobionts. And we have some control over them. And I could imagine in the near future, we're going to get better at using these viruses to help engineer healthier microbiomes. So on the front of talking about viruses, I think many of you actually know Forrest Rower, who I trained with in San Diego. And he would always tell us that cellular life exists so that viruses have something to eat. And you know, viruses are clearly the most biodiverse entities on the planet. There's huge numbers of viruses. They have big impact on determining which strains survive in a particular system, which microbes get to live there. Most of the genetic diversity is actually coming from the viruses. Now, before I get too far, I want to define bacteriophages. Bacteriophages are the viruses that infect bacteria. And this idea of phage therapy is the idea of using the viruses that infect bacteria as alternatives to antibiotics. So I don't have to tell this room that we have a crisis surrounding treatment of antibiotic-resistant pathogens. And so the idea is to use the phages as alternatives to antibiotics and to have a new way of helping to more specifically control infection. I like to think about this in the context of cystic fibrosis. And I've been part of a group in Colorado in Telluride that meets every other year. And we've built a guild model of the cystic fibrosis airway ecology. It was a great time to turn down the lights, actually, thank you. Because now you can see some of the detail in this painting, which was developed by Ben Darby, who's an art student in San Diego, in Forest Group actually. And I really love how this came together. And really the idea is that rather than trying to pronounce the complicated taxonomic names of different bacteria, you can think about each bacteria in this community as having a different role and having a different job in the community. And so, you know, this has been many years of brainstorming together that we've thought through the different guilds that, that participate in the cystic fibrosis airway ecology. There's a lot of anaerobic environments, so you're gonna have anaerobes, you're gonna have fermentation going on. The fermentation products are essentially like alcohol leading to cross-feeding for some of the other bacteria in the community. So you can even see a beer mug if you look closely in here. And you've got waste products being consumed, putrefaction, for example. So you can see a skull representing that in this painting. And so anyway, I'm happy to discuss this further. And really, I'm trying to get into the viruses. But I want us to be thinking about what the conditions on the ground are like when you're talking about trying to use a phage to treat an infection. So we think of the chronic airway infections in cystic fibrosis as having two main populations. One, in some ways, is coming from the oral cavity. You have one of the most densely colonized microbial communities on the planet living in your teeth and in your oral cavity. There's a lot of anaerobes, which actually, when I first learned that, it really blew my mind that anaerobic bacteria that would just not be able to survive a single molecule of oxygen are happily residing in our oral cavity despite all the oxygen in the air. I think that's amazing. And so you've got these anaerobes and facultative anaerobes that can handle a little oxygen living in your oral cavity. And then you, in the context of an infection, you often end up with opportunists, many of them coming from tap water that can do, you know, can colonize the airways. And they are exchanging compounds. So their proximity doesn't even really have too much impact on how much communication they have, because many of the molecules produced by one member of this community can be volatile and travel far. They can, if they're liquid, they can still, those molecules can still travel. So these communities are in contact in a lot of different ways that we might not realize. Now, I want to just point out that in the middle of all this, the viruses are busy evolving. So each person has a very unique community, and the viruses, again, are at the cutting edge of that. So there's a constant arms race between all the phages infecting the bacteria in this community. There's often potentially as many as 10 different types of phages infecting each microbial cell. So this is going on regardless of any ideas about phage therapy. However, currently, we're much more often using antibiotics, which are a blunter tool. And I would love to see in the next 10 years, or we'll see how long it takes, that we have more opportunities to do more targeted therapeutics. So phages that we've just been talking about, they tend to be more specific. So you can look at that as a real positive, that the phages tend to infect just one type of microbe. So you can have phages that will only infect a very specific type of Pseudomonas. They'll leave the rest of the community intact. They'll have less impact on your gut microbiome and your oral microbiome if you're using them to treat an airway infection. You can also view that as a downfall, because the fact that they're so specific means that it's actually quite difficult to find a phage to use for treatment. In fact, it becomes quite personalized. You have to go and literally hunt one by one for a phage that can help each person. That's how it is right now. Of course, that's something we're very interested in improving, and many of the people in this room have had a big impact on that, actually. So anyhow, this is kind of one of the motivations. Now, phages have been used for therapy for quite some time. In fact, this advertisement from Paris in 1936 has some familiar names that you guys have all seen before, I'm sure. So these are phages that could treat many of the types of pathogens that we all encounter today as well. And so that's so interesting that we were already doing this back in 1936. But then I also, in my classes, I've been teaching about probiotics in my microbiome class. It's an $80 billion industry, and there's a lot of crazy ads out there. And I was at a conference in San Diego a few years ago, and I was looking at this package, which was being advertised as a probiotic. And I look at the ingredients over here, and I see myovirus, cyphovirus. These are phages. So phages are actually also marketed as probiotics today. You could buy these on Amazon, by the way. These are actually E. coli phages, and their initial intent was to use them as a treatment for traveler's diarrhea. But that's actually quite difficult to make claims that are allowed, for example. And so they instead are more generally referring to them as probiotics, and they're generally regarded as safe, and they could potentially, improve your microbiome in a more generic way. But we have actually tried these phages in our lab to see if they're active. And we have found that these phages from that jar can infect E. coli in our lab. So that was kind of interesting. And so it's not just Paris 1936 where you can find phages. And in fact, in San Francisco, there's a company that sells phages to treat acne. And actually, it's a really cool idea. There's a lot of teenagers who are taking a lot of antibiotics, chronically taking antibiotics to treat acne. And so I love this idea, actually. And so these are Corynebacteria phages, is my understanding. And you can see they're calling them probiotics, but powered by phages. So I think this is fascinating. And they really make it look good, So this, if you have someone in your life who's looking to avoid taking antibiotics to treat acne, this could be an option. But it's been a really big year for those of us who have been working on phages. To me, it feels like a bit of a sea change in the number of cases that are adding up. This paper comes from 2 years ago and has 100 consecutive personalized phage therapy cases being represented. And my understanding is that there's another paper coming out in the very near future that has 200 cases. So we're starting to have some evidence to look at to assess these one by one personalized cases. Now, this is a very diverse set of cases that were in different clinics treating totally different pathogens. So, it's not a classic clinical trial, and there have not been successful classic clinical trials with phages so far. There's just starting to be some right now. And by the way, there is no FDA approved treatment using phages. These are each one a compassionate use exemption context. However, look at the big print over here. Clinical improvement and eradication of the targeted bacteria were reported for more than 77% of the infections. So, this is, of course, again, it's very different from a clinical trial, but it's interesting to watch this evidence piling up. And so, of course, we're very curious what happens in our own lab with the case that just started yesterday. Now, there's a lot of unknowns when it comes to phages. In fact, every time we hunt a new phage out of wastewater and sequence it, many times it's totally new, or maybe sometimes there'll be one example of it from across the world, which also blows your mind. How did those phages land in such different places? So anyway, there's really a lot of unknowns, which in some ways is inspiring. It gives us a lot of motivation to work on this. If you look at the number of sequenced phages from some of the famous pathogens, it's a surprisingly small number. These numbers are growing, so probably you could find more. I think it's easy to say that we need more phages. And actually, if you notice, Mycobacterium is the pathogen for which we have the most phages. And that is because of undergraduate efforts. So there's a Howard Hughes funded class, Phage Hunters, where the students are going out and hunting Mycobacterium phages. So putting energy into this is effective. So that's actually very motivating to see what a big impact that had. But yeah, I mean, if you had asked me where we would be looking to find treatments, I would not have guessed the sewer. And, I definitely believe in the miracle of tap water. I drink tap water, and usually it's on the purified side of that equation that I would be leaning in. But we actually have been collecting wastewater samples from all around Southern California. We have partners at the Southern California Coastal Water Research Project where they have been giving us samples as well. And during the pandemic, we monitored those wastewater samples with sequencing. And from 8 samples, we could monitor 16 million people, which is amazing from just 8 samples. So anyway, this photo landed in the CFRI newsletter. We took this photo to have a joyful and sewage related picture to share with you. And they really got into it. But here's the idea is the students get really into this. And it's actually been a wonderful undergraduate research type of project to set up. It's been very doable. It requires patience and expertise from the more senior students, that's for sure. I owe a lot of credit to Kayla Ragoza and Alicia Monsibais who are in this photograph you can see. But anyway, we've always got a lot of students in the lab who are up for giving this a try, where we get the raw sewage samples, incubate them together with clinical strains, and then look for plaques. And so plaques refer to these clear zones on the white bacterial lawn background. So in this plate, the white bacterial background just looks like the kind of white background. And every time you see a clearance zone there, that represents a phage. And it's been for most pathogens, it's been very doable to find new phages. And there are some exceptions, but overall, it's actually not the limiting factor, I wouldn't say. Our phages have occasionally been out in the world. Here's an example of some Enterococcus phages. I had one era of students who loved giving the phages old man short names like Bill and Ben. And because they were short, they could write them on the test tubes. And so anyway, these ones made it into a case with a recurrent bacteremia. And Enterococcus has been a focus in our lab for some time. So this was an interesting example where we were able to share the phages, and other groups have made use of them as well. And over the years, we had a lot of experiences where people would send us a clinical isolate asking if we could find a phage. And the answer was usually yes, but it was never fast enough to be able to help. Something would always happen. The person would get better, or the person would pass away before we actually could get through all the different steps that were involved. So about a year ago, and with motivation from Jessica Satcher, who's here at Stanford, and Paul Boyke's group, we decided to ask the infectious disease department to consider cases that were more chronic so that we would have time, so that if it took us a year to prep the phage and get through all these approvals, we would have time to do that. But it had to be a case where it really would help the person, and it was worth the risk, because there is risk in taking on a new treatment like this. And so that was about a year, well, a little more than a year ago, actually. And so since around that time, we've screened at least 10 different cases where those criteria were met. And we have quite a few phages against all of the different main pathogens that come in. So Enterococcus, Stenotrophomonas, E. coli, Pseudomonas, and Staph aureus, we have those ready to go. And then of course we can screen the wastewater. And so overall, we have actually in each case been able to find phages. That's interesting because we also have a lot of experience sending our phages around. And I mean, Daria is in the audience, and we're often sending our phages around. And I found this really fascinating just recently, this comparison of whether a phage prep would work in different labs came out. And there's only 22% agreement between 3 labs when they each try to see if a phage can infect. And that really tracks with our experience. It's been so interesting. I'll think of us as having like the world's best murdering staph phage, and we send it to a friend's lab and they can't get it to grow. Or I have a phage that I've referred to as a wuss for 10 years, and I send it to a colleague and they send us these beautiful plaques, and I'm like, what did you guys do? This is amazing. And sometimes we can learn too. Sometimes we get the tricks. Oh, you need to add this ion to your buffer. But the reason I bring this up at a evening talk like this is just to point out what an early stage we're at for figuring out how to even communicate about how to set our phage assays up. There's a lot of room for improvement there. Another area that I think is really interesting, and actually Daria is even on this paper as well, is in this study of Burkholderia, looking for phages that could infect Burkholderia, they actually took multiple isolates from each person in the study, which is such a good idea. It's typical that we will get just one isolate from the clinical microlab to represent a whole complex community. And so in this case, they had multiple isolates of Burkholderia from each person. And they found that a lot of the time, a phage could infect one of the isolates, but not the others. And so that's also something we have to consider. Anyway, I just thought this was an interesting challenge to point out and a motivation to collect multiple isolates from each person so that we can learn more about the whole community. But when it comes down to it, I can summarize our 10 cases in the last year to say that we were able to find a phage every single time. There were lots of different pathogens involved, but only for one of them how did we get all the way to the point of FDA review. And so we produced the phage in January. We got the third-party sterility and endotoxin testing done. We applied to the FDA. We got that approval in early June, and it took until yesterday to get the IRB approval to be allowed to start the treatment. So anyway, it's a bit logistical to share all of that with you, but it's been such an interesting experience and a big part of my life this year that I can't help but share it. And so, yeah, just more information about this case. You know, it's a MRSA that's resistant to pretty much everything. And we didn't have a phage when we looked in our own freezers, so we had to go and isolate a new one. And that on its own has actually been very interesting. So the phage that we proceeded with is called Ludmilla. And with the student who really made all this happen, who has great lab hands, struggled to isolate a phage. And then we had a new medical resident volunteering 1 week. And the first time she tried, she got this phage. And that's so interesting to me too. I'm sure there was some detail in how she did things that made it work. Or it could have just been that that particular chunk of wastewater had the magic phage in it, I don't know. But anyway, you can see very nice big clearance zones. This is a different type of assay where you drop the phages down. But if you see a clearance zone, that tells you that the phage is killing the bacteria. And we have now sent Ludmilla to other labs that have better curation of staph strains, and this phage is killing lots of different types of staph. So that's exciting. And here's Ritwick, who did all of the prep and visited Daria Van Tine's lab to learn how all the bureaucracy worked. And that's him on Wednesday with the phages in the cooler. He even had a fresh lab coat in the drawer to put on before he took them out of the fridge. So he really thought of everything. And so that really made us feel that we were getting a little closer to making phage therapy happen. It felt not quite so out of reach. And that's kind of a theme in some of the projects we've gotten to do in the last few years related to phage therapy. Just very briefly, I want to share what it's been like for us to try to resurrect this century-old protocol called the Apelman's protocol, where if you have a recalcitrant bacterial strain that none of your phages can infect, you try an experimental evolution approach in the lab where you mix phages that are very effective and strains that are very susceptible together with the recalcitrant strain, with the hope that as you're propagating, there'll be a new mutant that arises that can then infect the strain that was recalcitrant. And it doesn't work every time, but we've actually had some really interesting success with it. And we have a lot of plans to use this approach, working with phage-resistant mutants and trying to work towards building phages that are able to overcome that. And so in this example, these are Enterococcus strains. And oh, I have a pointer here. That might be a better way to figure that out. So here you can see the ancestral phages that we started our experiment with. We're having, this is a summary showing you the percent of the strains that are suppressed. And you can see that the ancestral strains are having kind of middling success against many of our Enterococcus. But as we evolve them in different combinations, we get more and more success. And so that was very interesting to me. It didn't extend from from one type of Enterococcus eficalis to another Ephesium, but within subtypes of Eficalis, we see much greater suppression by these cocktails. So that's, to me, one direction to take for improving the capacity for phages to more generally infect their hosts. So that's just a real quick taste. However, another way we're hoping to give our phages an edge is by changing the molecular environment. And so that's what we were funded by CFRI to work on. And I'm going to share some of our results in that direction for the last part of my talk. So we were talking about the ecology of chronic airway infections, and you have these two main communities, the oral anaerobic type and the more opportunistic pathogens that can come to dominate the airways in a chronic infection. And you have exchange of volatile compounds. Sometimes those molecules can alter the microbial physiology. And this is how we first got into it. During my postdoc, I was even collecting breath samples to look for the fermentation products in the breath that actually, many of them come from microbes that you think of as oral microbes, but they of course can expand their territory in the context of an infection. So this was one reason we got interested in these kind of molecules, thinking about how the fermentation products from the oral cavity, for example, can impact the physiology of the chronic infecting pathogens. And of course, we've always got the viral individual evolution going on in each person. One of my students made a really beautiful painting to represent this, where she called it Smelly Night rather than Starry Night. And these little popcorn colored flavor bombs are not an accident. That's actually because one of the main molecules that you detect in the breath of a person who has a chronic airway infection is butanedione or diacetyl. It's actually the molecule that gives microwave popcorn its flavor. And it's a very toxic molecule. They actually have to regulate it. There were examples of factory workers who developed bronchiolitis obliterans after intense exposure to this molecule. And now I do not mean for this to discourage people from eating microwave popcorn, because the way most people consume it, it would not be a problem at all. There was one example of a guy who ate 2 bags of microwave popcorn every day for decades and inhaled deeply every time he took it out of the microwave. So that's an extreme case, and that did actually damage their lungs. But overall, it hasn't been a problem. But I mean, there's an amazing diversity of molecules, most of them unnamed, unstudied, but there's quite a few that we know a lot about that are part of the evolutionary history of these microbes that are in the infection. So that's the background of how we got interested in this in the first place. But of course, you can have a lot of strategies for improving phage activity. You can try coming up with molecules that will improve the efficacy of antibiotics or phages. I mean, this strategy is being used for antibiotics all the time. So really the idea is to layer this idea to help with the phages as well. You can try combining the phages into cocktails. We talked about using phage evolution to steer the phages towards having broader host range or other properties. Anyhow, you see the idea here. We talked about these small molecule adjuvants that could disrupt biofilms. They could alter the bacterial phenotype. The more we learn about it, the more I learn of all the different ways that this could have a big impact. Some small molecules will end up feeding staph cells and changing the way that they build their cell membranes. Well, that's going to definitely impact how phages can interact with them. So there's a lot of really interesting potential mechanisms here. And we started out with some of the favorite molecules that had emerged from breath studies, like 2,3-butanedione, and actually fatty acids were a category that we got really interested in. And Sage Dunham is in the audience here, and a lot of this is his work, and I really should be thanking him. But we had a lot of fun thinking about this. Fatty acids have a lot of potential. They're generally regarded as safe. They are, we're all exposed to them all the time. They're produced by anaerobic fermentation. This is a collection of fatty acids starting from C1 up to C9. So they're quite volatile when the carbon chains are smaller, and then they start to get more difficult to work with and solubilize in the lab. But so far, these are ones we've been able to work with. So they're present throughout our bodies at different concentrations. Some of them are they're known for being involved in quorum sensing. And some of the antimicrobial properties of the longer chain fatty acids have been known for a long time. Basically, in a way, we're talking about a version of soap. So that we know is antimicrobial. But for example, here's a treatment that we learned about, and I'm glad we're not eating while I'm talking about this. And so this is a treatment, a butyrate enema that is sometimes used to help in the context of inflammatory bowel circumstances. So butyrate, for example, is a big member of it's a really important molecule in the gut. It's one of the main fermentation products. When you eat fiber, you are fueling the anaerobes in your gut to make butyrate, which is the favorite fuel of the cells lining your colon. So when you eat fiber and you give those anaerobes their favorite foods, the fiber, they will produce butyrate. And then the cells lining your colon have a lot of interesting phenotypes that are critical to health. So by eating fiber, you reduce your risk of cancer. You have tighter junctions connecting those cells that line your colon, so you protect yourself from leaky gut, even from sepsis. So, many of you have heard me talk about fiber before, so I could definitely get into that if anybody wants to ask more. But anyways, another way to get this butyrate to the gut is in the form of an enema, which we were amazed to learn is an actual treatment that is used. I did hear from nurses involved that it doesn't smell very good, but apparently it has some efficacy. So these ideas have been around. Here's a paper bringing up the idea to use short chain fatty acids as treatments in the lung. So, the fact that there's a little bit of history here is encouraging. So we took a look at this chain of short chain fatty acids starting from C1 and getting bigger. So starting from the more volatile ones and getting bigger. It's fun to think about all the different properties. Sage put this table together, and I was fascinated to see what the odor was of the different molecules as the carbon chains grow. So that's quite interesting. And so we started testing these. We started testing them on their own, and then eventually with the phages. So I'm going to share some of the data from how we've been testing them. I wasn't sure if we would all be eating while I gave this talk, and I was going to have to really move past that kind of quickly, but I'm glad that it. Yeah, and I don't know if I personally have identified those odors myself. I mean, acetic acid, we all know what that smells like. So we actually started with Stenotripomonas multifida. Actually, during the summer of 2020, we received a patient isolate of Stenotripomonas, and many of the labs that hunt for phages had told us that they had not been able to find any stenophages. And for reasons I'll never know, the Escondido wastewater we had just always succeeds. So that got us really interested in working with Steno, which already is an underdog that I loved working with. In fact, my very first grad student, when we had an isolate in the lab that we thought was Pseudomonas, we were looking at the sequence data, and it was not Pseudomonas, it was Stenotripomonas. So actually that's the main reason we first started working on Steno, which is kind of interesting. But so here we have a nice collection of stenophages. So we wanted to see if we could use these adjuvants with Steno. Steno is a gram-negative opportunistic pathogen that is often found in the CF airways. It's often highly resistant to available antibiotics and forms biofilms. And so it was easy to justify the need for new strategies to target Steno. And so, as I mentioned, we have a collection of stenophages that has been growing. And here you can see 3 of our main characters that we've put a lot of energy into characterizing. And so, we were interested in seeing if we could improve their activity. In our collection of Steno, we have about 100 Steno strains in our lab. one by one, these phages can infect about 30% of them. So there's real room for improvement. Okay, so the first thing we tried was just to concentration gradients of the different fatty acids against Stenotripomonas. There's a lot of considerations here that we really had to think a lot, you know, in order to watch improvement in any kind of assay. You need to have room for improvement. So you have to have growth that's robust enough that you can actually watch a reduction in the growth. And when you're dealing with strains that have all different growth rates, it's actually amazing how much troubleshooting we had to do just to set these assays up. So that was actually pretty interesting. Over a couple of years of troubleshooting, we have a nice way of assaying things now. One of the first things we noticed was that many of the higher concentrations of these more volatile, smaller short chain fatty acids were effective in killing or reducing the growth, at least, of Stenotripomonas. So these are increasing concentrations, and you can see a reduction in growth. This is a measure of growth of Stenotripomonas. And so that was interesting. However, we also know these molecules are quite acidic. In fact, we had even written papers about how Stenotripomonas has a very strain-specific but strong response to pH. So in one of our lab meetings, we had a discussion of high school chemistry, and we realized that we should actually be doing these experiments in a way that keeps the pH neutral. So we managed to do that by using the pH neutral fatty acids, like an acid and its conjugate base. And so when you keep the pH neutral, actually we don't see such a big reduction in growth from the fatty acids in that case, from the short chain fatty acids in that case. But as the carbon chains grow, and then you start to see a pH independent growth reduction from the medium chain fatty acids. And slow me down if anybody has any questions here, for sure. Okay, so now I've shown you what goes on with the fatty acids on their own, but really we're looking to use these molecules as adjuvants to help the phages, right? And so now I'm going to show you some data where we are making a comparison of phage and fatty acid to watch the combination effect on the growth of the bacteria. And so you can see here, we have concentration gradients of phage and of the fatty acids. And then we can look for additive or synergistic properties here in these what we call checkerboard assays. And so if you want to have a look at it, you know, keep in mind up in this corner is where you're going to see both high phage and high fatty acid concentrations. Okay, so one of the initial things we found was that with this particular phage and the C8, the 8 carbon chain length acid, octanoic acid, we see really interesting synergy. And so you can see that in this assay here. I'm going to show you this data in a few ways. So you can see the growth reduction with the octanoic acid and the phage. And then we've actually been building different types of interaction metrics. And you can see that there's a favorable interaction between the phage and the molecule. So this requires you to have room for improvement on both sides. If you started out with a phage that murders everything, you have no room to learn if the molecule is able to help. But in this case, this is actually one of our slower phages. So it's turned out to be a good testing ground for the synergy. And so here you can see a clear beneficial interaction by the combination of the C8 octanoic acid and this phage. So zooming out a bit, here I can show you across all of the different carbon chain lengths what goes on. And so for the medium chain fatty acids, we have a pH independent and blue, so positive, as in synergy type of interaction, suggesting that the combination is effective. So it's interesting to have this possibility. We've been thinking a lot about the mechanisms. And we've also been interested to know if this actually is killing the cells. And yes, indeed, if we take the cultures after exposure to these combinations of phages and the molecules, they don't grow. So it appears to be bactericidal. And so we've now expanded this to looking into many more strains and looking into new conditions. We were very interested to use the cystic fibrosis sputum media that's been developed by Marvin Whiteley's company, Synthbiome. And so now I can show you in many strains of Stenotrichomonas and in different media types, we do see some efficacy. So we need, you know, I'm just going to help you guys walk through this data. So I'm showing you bacteria only, and then the addition of a medium concentration of phage and a medium concentration of fatty acid, hopefully giving us room to see improvement. And so here's where you see the combination in this row, the phage and the fatty acid. And now you're going to get to see across about 20 strains of Stenotrichomonas that we see quite an improvement in growth reduction when you have the combined phage and fatty acid. So this is with our slower growing, this ANB28 is one of our slower growing phages. So now you might be interested to know, well, what happens with other phages when you try this against the same collection? And so now KB824, for example, is our fastest murdering photovirus kind of a phage. And you can see that actually we do still see positive combinatorial effect when we have both the phage and the fatty acid. Now there's of course exceptions, and you could stare at this data for years. We have in our lab meetings, it's very fun to look at. For example, this SM50 is a very slow growing bacteria. And so it's actually hard to come up with a way to see improvement because it doesn't grow very much. So I wouldn't actually say that this means, you gotta pick your circumstances to learn as much as you can about these interactions, which of course is really interesting to think about how to make a universal treatment when you know that so many things are condition specific. However, speaking of conditions, we have also tried this in the synthetic cystic fibrosis media, which was developed based on mass spec data from sputum samples. So it's trying to make a molecular composition that represents sputum. And so you can see that we actually still do see a synergistic combinations with the phage and the fatty acid across many different strains of Stenotrichomonas. Okay, so my last thing I want to share with you is that we also tried this in gram-positive bacteria. So I've only told you about steno, a gram-negative, and so now we've also been trying this in some of our gram-positive strains. Here's a collection of more than 100 clinical isolates of Enterococcus that we've been working with. Actually, I showed you our evolution data, so we had this all set up so we could test it out with Enterococcus. And you'll notice that, the phage alone have kind of this spotty impact. The octanoate, the 8-carbon chain that we chose, has a pretty unimpressive impact on growth, but the combination is really knocking out a lot of these strains. So that to me is fascinating, that it's working under totally different cell type. Gram-positive cells are so different. And I can see that I should wrap it up, but I will just quickly share that we have also tested this in the strains of Staphylococcus and Pseudomonas that we have in the lab. And of course, there's some strain specific outcomes, but overall, we are finding that these additive effects are working in many different strain types. And so you can see it's kind of concentration dependent, but you do see blue synergy and white additive effects emerging in both Staph and Pseudomonas. Okay, so thank you very much for your attention, and I will share my particulars with you.